Hello, welcome everyone to round 2 of the ReachOut Science Slam semifinals. Now, before we get started, if you would like to access captions for this event, you can find a link to live captions in the YouTube description below, and we're also going to put up a QR code here on the screen that you can use to access those captions. All right, well, welcome again to the round 2 of the ReachOut Science Slam semifinals. I am Megan from the Museum of Science. My pronouns are she and her, and I am so excited to be your host tonight. Now, these events are part of the ReachOut Science Communication Challenge, a Museum of Science program designed to train scientists to share their work with public audiences and then put that training to the test through some friendly competition here at the ReachOut Science Slams. Now, at each of our 3 semifinals events, 2 slammers are selected to move on to the big finals event on May 4th and compete for our top prizes. Now, last week, our 2 slammers chosen to advance to the finals were Jolene de Lima from the Beacon Center for the Study of Evolution in Action. She was our audience pick. And also Giska Ramadita from the Center for Engineering Mechanobiology was our judges pick. So congrats to Jolene and Giska. We will see them again at the finals on May 4th. Now tonight, our second group of semifinalists will compete, followed by one more semifinals round on April 20th, and then the big finals event on May 4th. So make sure you tune in to all of these exciting events. Now, again, tonight, 2 of our slammers will be chosen to advance to the finals, and 1 will be selected by you, our audience. So stick around, and we will tell you how to vote for your favorite science slammer towards the end of the program. Now, the other finalist tonight will be chosen by our incredible panel of expert judges. Joining our judging panel for this semifinal round, we have 3 outstanding leaders in science communication who are here to lend their expertise and mentor our slammers tonight. Raven Baxter, also known as Raven the Science Maven, is a molecular biologist, expert science communicator, and has been featured in Fortune magazine's 40 Under 40 list for 2020. Melissa Cristina Marquez is a shark scientist, TV presenter for National Geographic and the Discovery Channel, and author of the upcoming book series Wild Survival. And to round out our judging panel, Jorge Cham is the creator of the webcomic Piled Higher and Deeper, or PhD Comics, and he's co-creator of the PBS show Eleanor Wonders Why. Welcome to all of our judges. All right, well, welcome, Raven, Melissa, and Jorge. We are so excited to have you all here. Now, are you ready to hear from our slammers?
Yes.
So excited.
Yeah.
I'm ready to be slammed by science.
Wonderful.
And have fun.
Our ReachOut Science Slammers come from all over the country and are each part of one of the 12 National Science Foundation Science and Technology Centers. These are interdisciplinary research centers, which means a bunch of different kinds of scientists come together to discover something new about the world or solve some big challenges. They're working on all sorts of areas like microbiology, artificial intelligence, evolution, quantum physics, cell biology, computer science, and more. All right, now today's slammers come from 3 different centers. We have 2 slammers representing the Center for Cellular Construction, which aims to develop a new engineering discipline that will allow us to design and build cells and tissue with special structures that could one day serve as living factories and building blocks for better and more sustainable materials and products. Now from CCC, please welcome Anamika and Megan. All right. And next, we have 2 slammers from the Center for Dark Energy Biosphere Investigations, which discovers microbial life within rocks and sediments and studies how they affect global cycles that keep our Earth healthy. From CDebbie, we have Rachel and Sabrina. And finally, we have 3 slammers from the Center for Biology with X-ray Free Electron Lasers, which uses X-ray lasers to watch biomolecular machines at work to better understand how life works at the molecular level. From BioXFEL, we have Tech Nursing, and competing as a team, we have Anna Sophia and Gabriella. All right, welcome to all of our slammers. We are all very excited to hear from all of you. Now, if you at home would like to cheer on our slammers, you can send a comment at any time by going to pollev.com/Museum of Science, or you can text Museum of Science to 22333 and then send your comment. And we'll keep that information handy on the screen for you. But please note that right now, we're just taking some comments to cheer on our slammers, so don't cast your votes just yet. We'll let you know when the audience voting opens a little bit later in the show. All right, is everybody ready? Let's get started with our first slammer of the evening. Our first Science Slam contender is Anamika Agarwal, who is joining us from the University of California, San Diego, where she is connected to the Center for Cellular Construction. Welcome, Anamika.
If I ask you, what is the one thing you will always remember from your biology class? Your answer is probably going to be mitochondria is the powerhouse of the cell. These little bean-like objects in our cells are single-handedly responsible for producing the energy that our cells need to be alive and running. But did you know which organ of our body uses the most energy? It's not our muscles. It's not our heart. It is actually our brain. Every cell of our hungry brain needs not one, but hundreds of mitochondria that are working extra hard at their jobs of energy production. When we look at brain cells, which are also called neurons, under a microscope, we see these tiny little mitochondria that are rushing around the narrow freeways of neurons to supply energy to each part of the brain. Now, their traffic patterns are the most amusing and sometimes seem to make no rhyme or reason. These mitochondria sometimes move around, but they spend a lot of their time chilling about in traffic jams. They also seem to be saying hi to each other in the middle of the road. This makes scientists like me curious. Are these brain mitochondria doing their jobs at all? To answer this question, scientists put on glow-in-the-dark tags on the mitochondria to follow them along a neuron. They found out that when we observe interactions between mitochondria, they are actually helping each other out. In fact, the healthier mitochondria can sense that their friends are sick, so they can transfer some of their power-making juice to them and make them strong and capable again. Isn't that pretty smart? In fact, even the traffic jams seem to have a reason. Whenever and wherever our brain generates an electric signal to transfer information, it needs tons of energy that the mitochondria need to supply. Now, what happens is, coincidentally, these same electric signals also happen to be red traffic lights for the mitochondria, who have braking molecules that can see these red lights. So these mitochondria are jammed exactly where they need to be. They are as smart as any self-driving car out there. Now, mitochondria are pretty awesome, but they are just one of the few elements that form the dynamic cityscape of the cell. Each teeny tiny city within the cell has not just fuel centers, but also freeways to move things along. It also has its own administrative and planning center, and even its own recycling center. At the Center for Cellular Construction, we marvel at the intricacies of this well-planned city and use some of its technologies to solve our own real-life problems. Thank you.
All right, let's hear it for Anamika. Thank you, that was wonderful. Now let's check in quickly with our judges. So what did you all think? Raven, would you like to go first? What did you think?
Sure. I really loved the visual representations that you gave, and it helped bring us along to what your thought process is as you think about the science and how you want us to understand it. I thought that was a really great way to talk about what you were talking about. Good job.
Thanks. Melissa and Jorge, anything to add?
Yeah, I thought it was
I
really
thought
well
you're
done. Sorry, Jorge. I thought it was really well done. The visuals were really interesting. The examples and kind of like the analogies you gave, like them waiting in traffic and waving hi to one another, that was really cute, and kind of gives you a better understanding of what you're talking about. So great job.
Yeah, I thought I really liked your energy and your enthusiasm, and you seemed really excited about the topic, which made me excited. That was pretty cool. Yeah. I think maybe it would have been nice to like maybe hear like a specific, go a little bit deeper into like maybe a specific question or problem that you're maybe tackling or that you find interesting. But overall, yeah, it was fun, and the graphics were fun as well.
Thank you so much.
Great. Thank you so much to our judges. Awesome job, Anonymika. And now let's take a look at slam number 2. Next up, we have Rachel Weisand, a student at Texas A&M University and part of the Center for Dark Energy Biosphere Investigations. Take it away, Rachel.
Have you ever been to a beautiful place like this? Imagine you're driving down the coast when suddenly you find a place to pull over and really explore where the land meets the sea, a wetland. So you get out and you start to explore, and a bunch of senses hit you all at once. You hear the sound of the waves hitting the shore, as well as the humming of the insects around and the birds flying above. And you see these beautiful wetland plants, like these mangroves, standing tall in the mud that you're walking on. And speaking of that mud, it's really icky and gooey, and you really have to be careful where you place your feet because you're afraid you're going to fall over, and it smells so bad. Speaking of which, why does it smell so bad? It smells like rotten eggs. Well, so what's happening is there's a bunch of beautiful things that we can see in this wetland, the birds, the fish, the insects, the plants, but there's a bunch we can't see. There's these teeny tiny microscopic organisms living in the mud that we're walking on. So in the mud, there are these organisms, microbes, that are creating energy, and they're actually producing that rotten egg smell. So like we breathe in air and produce carbon dioxide, these microbes are breathing in something in the mud and producing hydrogen sulfide, or that rotten egg smell. And so while we can't see these microbes with our eyes, we can actually prove that they're there. So what I'm showing now is this cup of mud. And in this cup of mud, we've attached a bunch of wires, we've attached this light bulb, and we are showing that these microbes are producing this energy, and we're using it to make that little light bulb go. Yes, they're working, and we're just exploiting it to make the light bulb work. And that's really cool. And that proves that we've got this microbial community working really hard. So what does that mean, microbial community? Well, there's not just one type of microbe that's producing this hydrogen sulfide smell. There's actually a bunch of different microbes that are working hard together. And what's really cool is they like to share. So sometimes one microbe can produce something and another microbe can repurpose it for its own energy uses. So they're good neighbors, they like to share, we love it. So what happens for that microbial community? And what does that mean for our overall wetland? So at our lab, we're really interested in looking at how the microbes affect our wetland health and what they're doing within the sediment or the mud. We like to see the reactions they're doing and how they're interacting with each other and how it might change over time or where they are in the mud. Are they close to a plant? Are they under the water? Are they being exposed by a bunch of things in the mud like worms and insects? So next time you're in a wetland like this, remember that that mud isn't all gross. It's actually really cool. It's a bunch of microbes working together. Thank you.
All right, let's hear it for Rachel. Thank you, Rachel. That was great. So now let's hear from our judges again. So Melissa, what did you think?
I really love, Rachel, how you brought the wetlands to life through all of our senses, our sight, hearing, touch, smell. I thought you did a really great job creating a story, weaving scientific facts into it, telling us how it impacts us. Great job.
Great, Raven. How about you?
I love that as well. I think that one of the hardest things to do as a science communicator when you're talking about microscopic things is really getting people to connect with the material and when they can't see it. So it was really important. I felt it was really important that you did paint that picture. Like, what are we smelling? What does it feel like to be in this environment? And then the props that you had to piece together different parts of your communication. It just overall made what you were doing a very strong piece of work. So congratulations, you did a great job.
Thank you.
Great, thanks, Raven. Jorge, anything to add to that?
Yeah, just to echo what Raven and Melissa said, I really sort of liked your presence, and you're very relaxed and very sort of engaged with the audience and the camera. That was great. And also, I found myself really engaged with like these, like Melissa said, you know, you were engaging our senses and getting us to think about smells and sounds, and that was pretty cool.
Thank you.
Great. Well, thank you so much, everyone. Great job, Rachel. And now let's move on to slam number 3. Our next slammer is Tech Nursing Mala, representing the Center for Biology with X-ray Free Electron Lasers, and he is a student at the University of Wisconsin-Milwaukee. Over to you, Tech Nursing.
Nobody likes being sick, yet we all have been sick at some point in our life. And every time we are sick, there's a battle going on between our body and the disease germs. And this is a battle we have to win. We must, because we recover only when we win. That's why, over the course of history, we developed medicines. They are our weapons to win this war. But diseases are getting harder and harder to cure. Over time, they also develop their own defense system that makes our medicine absolutely useless. Doctors sometimes prescribe a new medicine or increase the dosage in response, but that is not the complete solution. Because when we get new medicines, the germs can also evolve to develop new defense. And we get more, they get more. They will only make them super resistant. Take tuberculosis, for example. We are already at the 5th iteration of antibiotics. Although this disease is curable, the bacteria are very resistant against our medicine, causing over 1 million deaths every year. So we need a new strategy. And one such strategy is to study their defense system. And that is precisely what scientists like me are doing. We are a part of BioXFEL Science and Technology Center, and we have collaborators all around the world tackling the same problem. Together, we use powerful X-rays to study the biological molecules. Because these are super small, only X-rays can see them. So using X-rays, we studied this protein. It is produced by tuberculosis bacteria as a part of their defense system. And this protein literally breaks our antibiotic in half, making our medicine totally useless. So no amount of medicine will work as long as this protein is there. So by studying how this process occurs, scientists developed a new medicine which looks very similar to our antibiotic. We can now trick this protein to grab the new medicine instead. However, this time, the medicine gets stuck and the entire protein becomes useless. And without any active defense, the bacteria are prone to our original antibiotic. And we win this war. Hey! Okay, this is one of the reasons we take multiple medicines these days. One, to disable their resistance system, and one, to kill them. This idea can be applied to other diseases as well. For example, this beautiful heart-shaped protein is what makes coronavirus so contagious. Once again, if we can find a medicine that gets stuck inside this heart, the virus cannot multiply, and we win this war against pandemic. This is one of the many ways BioExcel is contributing to the community, and I feel incredible to be a part of it. Thank you.
All right, great. Let's hear it for tech nursing. Thank you. Awesome job. All right, now this time, Jorge, what are you thinking?
Well, as a cartoonist, I was and I'm in the animation business as well. I was really impressed, Tech, by your graphics and your animation and your timing. I thought that was really sort of cohesive and fun and really just very clear, like how the story unfolded and how, you know, all the twists and turns in the story kind of they were all really well animated and illustrated. I thought that was really well done. Congrats. And just in general, you were very relaxed and very cool and very confident and engaging, and that was great. Yeah.
Great, thanks, Jorge. Raven, how about you? What are you thinking?
I love your enthusiasm, especially your woohoo in the middle of your talk. You know, it just reinvigorates like my attention and interest in what you're talking about. And I mean, if there's anything I've learned from being a science communicator, it's that if you're excited about what you're talking about, even if who you're talking to, if they don't even understand what you're saying, they're going to be more likely to ask you questions because they want to be as excited as you are. They don't want to miss out on what you're excited about or the joke or anything. They want to be involved in that. So definitely good job on your enthusiasm. Always don't stop being enthusiastic about your science. It's awesome to see.
Great, thank you, Raven. Melissa, anything to add?
Yeah, I mean, Jorge and Raven definitely said it. I think it's great that you showed that passion and enthusiasm you have for your topic. It was just really great timing throughout the talk as well, both with your pacing and adding those little bits of excitement in them. I thought it was absolutely great. So yeah, keep that up.
Thank you, guys.
All right, thank you, judges, and thank you, Tech Nursing. Great job. You did wonderful. And now it's time to hear from Slammer number 4. Heading into the second half of our slams this evening, Megan Chong is our next contestant, and she is a student joining us from the University of California, San Francisco, and she's affiliated with the Center for Cellular Construction. Welcome, Megan.
We are all made of about 30 trillion cells, and every day 2 trillion of them do something called cell division. The purpose of cell division is just to make more cells as your body grows, and later to replace cells you've lost. So that's something like 700 trillion divisions happening in your body every year. And today, I want to talk about the challenges cells face every time they divide. Most of you have heard of DNA. Sometimes called chromosomes. DNA gives your cells instructions on how to make you. And it's really important that each new cell you make has a full set of those instructions. One chromosome looks something like this. And just before a cell divides, it makes copies of all its chromosomes and sticks the copies together like so. And the key challenge in cell division is to make sure these copied chromosomes are split evenly into 2 new cells. So let's take a look at a dividing cell. This cell is outlined in purple, and it has its chromosomes at the center, and something called the spindle is highlighted in green. And as I play this movie, you'll see that the spindle is just a structure the cell builds that attaches to chromosomes and pulls them apart as the cell divides. And building that structure well is really important for your health. Failing to separate your chromosome pairs or letting cell division get out of control can lead to diseases like cancer. So how do cells actually solve this technical problem of engineering a spindle that can divide your genome evenly every time? You might think that, like an engineer, they use some sort of blueprint or detailed instructions like you'd find in a Lego set that say, attach chromosome 1 to both sides of the spindle, now chromosome 2. Instead, the cell takes a much more winding path through division, and that helps make chromosome separation more accurate. You can think of this like solving a maze. If I plopped you down in this maze with directions that told you which way to go at each fork, you could cut a clean path through. But then if you tried to solve a new maze with those old instructions, you'd get stuck pretty quickly. So what if instead I could give you a simple trick or a rule to follow that would get you out of any maze? You might have heard of the right hand rule. You can find your way through a maze by keeping your right hand on the wall beside you as you wind your way through. Now, your path is a lot more roundabout, but you'll definitely get out. So back to our dividing cell. As cells build these spindles, sometimes they make some wrong turns. Sometimes both copies of the same chromosome pair attach to just one side of the spindle. Other times, a chromosome pair doesn't touch the spindle at all. In this case, the rule is that chromosomes send a loud signal saying, "We're not attached," and the cell waits for the spindle to correct its mistake. By understanding more about the signals cells listen to build the spindle, we can improve our understanding of diseases like cancer and design new treatments. But we can also learn about the general rules cells are using to build complex structures. And with those rules, we hope to begin engineering better versions that will avoid mistakes altogether. Thank you.
All right, let's hear it for Megan. Thank you, Megan. Great job. All right, now, Raven, what did you think about that?
I really enjoyed it. I'm a cellular biologist, so and I used to produce those types of images, and it's really interesting. I definitely would have loved to hear more. I'm like sitting on the edge of my seat, like, oh, tell me more. I can tell you know so much about your subject area, so I'm definitely looking forward to seeing more from you.
Great, thank you. Melissa, what did you think?
I thought it was great. I really liked how you got your message across with multiple props, such as the video, the animations, the physical props. And I thought your communication style is spot on. You know, you're relaxed, you've got good pauses, your funny jokes, a little bit of sass mixed in there. So yeah, great job.
Thank you.
Awesome. Jorge, how about you?
Yeah, same here. I like that a lot. And I also wanted to know more, even though I'm not a molecular biologist by far. But it was really interesting. And like Melissa said and Raymond said, I thought the visuals were very kind of visceral, like that movie of showing the chromosomes getting split. That was pretty fascinating. And then sort of the way you echoed that with the props and really kind of sort of drove it home and made it feel like personal. Like, you know, you made some references to like our cells and how there's a bazillion of them dividing in our bodies. I thought that connection, making us kind of feel that problem or feel the phenomenon was pretty cool.
Cool, thanks so much.
Great, well, thank you, judges, and thank you again, Megan. Great job. And now let's move on to slam number 5.
Now, we'll hear from our first and only teen entry to the ReachOut Slams. We have Ana Sofia Santiago Ruz and Gabriela Diaz Figueroa representing the Center for Biology with X-ray Free Electron Lasers, and they are students at the University of Puerto Rico. Take it away, Ana and Gabriela.
Oh, hi, Gabby. I just read a tweet that said that Michael Jackson was going through vaccine trials when he died. Can you believe that?
Wait, what? Are you sure? Wait, didn't we learn about vaccines a couple semesters ago?
Girl, I can't remember what I had for lunch yesterday. Let me pull out the presentation. Oh, yeah, I remember. I remember this.
Yeah, a vaccine is something that we put into our bodies that helps us from getting a disease that makes us feel sick or yucky. Believe it or not, vaccines contains little bits of germs. It seems kind of weird that you're trying to get yourself from getting sick by putting germs inside your body, but the germs are either weak or they're dead, so your body gets to prepare for when the real big guys come.
That's cool. And another type of vaccine is the mRNA vaccines, and they have been studied for decades. Basically, mRNA is DNA written in a different way or in a different language, if you will, and they contain instructions for certain important processes in our bodies. The mRNA vaccines for COVID-19, as example, contain information that helps our immune system develop tools and armor for combating this disease. But how do we know all of this? It's called structural biology. You remember this?
Yeah, girl, structural biology. That's the study of how biological molecules are built. Using a variety of imaging techniques, scientists have viewed molecules in 3 dimensions. This has helped them learn how these molecules are assembled, how they function, and how they interact with each other. Researchers have learned how the million trillions molecules that are inside of our cells behave and how they keep us healthy. We can use COVID-19 for an example. Scientists were able to uncover their 3-dimensional structure in just days. Can you believe that?
That's so cool. And BioExfil is also a cool laboratory that studies biological structures of very small molecules. And they virtually zoom in to understand how they work. That's so cool.
Yes, do you remember Dr. Daria Marchani? She used to be a grad student from our lab, and right now she's currently working on one of the COVID-19 proteins. She's really zooming in on that to understand how it works as a potential vaccine target. And
awesome.
Anna, didn't you do a BioExcel internship and studied what was it, structural vaccinology? Can't remember that.
Yeah, that's such a big word, but it's actually really cool. Just imagine an unstoppable copying machine. We are trying to hit that cancel button on the virus replication. The virus copies uncontrollably inside of our bodies, and we are just trying to find ways to stop that. So we hope this helped you guys understand how vaccines work with the help of structural biology. Remember, guys, to always seek the truth and be objective. Thank you.
Thank you.
All right, let's hear it for Ana Sofia and Gabriela. Great job. Thank you. All right, let's start with Melissa this time. What are you thinking?
I loved how creative this talk was with both of you guys playing off of each other's energy. It felt like a conversation that you can learn from. And it was great that it was current to humans currently because of COVID, but also vaccines in general are usually something that's talked about. So great job.
Great, thank you. Jorge, what are you thinking?
I agree. It was fun. It was definitely I feel like a creative risk for you guys to do it together. But I thought it was fun and not something you see a lot. Sort of like a little skit almost that you guys wrote and were playing out. That was pretty fun and pretty cool to see. And overall, yeah, it just it seems very relevant. And I like how you were sort of making connections to people, real people, and things that were happening right now. That was great.
Great, thank you. And Raven, how about you? Anything to add?
Yes. Oh, where do I begin? I think what I love the most was your confidence. Honestly, it takes a lot of confidence to do what you do. And I feel like you have to know or own your confidence as a science communicator to be able to make a skit about science communication, or demonstrating science communication rather. I loved how you both really were unapologetic about your personalities as people. You weren't I didn't feel like you were trying to play a scientist. It just felt like you were two scientists just chilling. Well, one of you was pretending not to be, but, I think, but it just really felt down to earth. And I think it was very refreshing for me to see that. And I think that would connect with a lot of people who are often missed in science communication. So great job.
Great, wonderful. Thank you, judges. And thank you again, Ana Sophia and Gabriella. That was fantastic. All right, and now let's take a look at our final slam of the evening.
And last, but certainly not least, is Sabrina Alcasis, a student at the Massachusetts Institute of Technology and Woods Hole Oceanographic Institution, and she's affiliated with the Center for Dark Energy Biosphere Investigations. Over to you, Sabrina.
A lake so toxic to life it seems almost extraterrestrial. This isn't sand that you see, it's mineral deposits. Nothing should live here in these waters containing a huge amount of chemicals, like baking soda, that make it seemingly inhabitable for life, right? Wrong. There are tiny, tiny microbes called archaea, which populate this lake. Now, they don't breathe air like you and me. They breathe hydrogen. They don't eat hamburgers and mac and cheese like you and me. They eat carbon dioxide. They live outside the conditions considered normal for life, almost like aliens right here on Earth. In my lab, I began simulating the conditions of this lake that I showed you earlier in this little vial. That's right, folks. In this vial, there are billions of microbes. Billions. But why bother? It's cool and all that I'm growing these, but why go through the trouble of growing them in the first place? Well, I want to bring your attention to this picture here. I'll give you a few seconds to come up with a guess of what you think it is. Okay, do you have your guess? Well, if I had to guess your guess, I would guess that you think it's the night sky or outer space, or in other words, really, really big. It is in fact not really big. It's the opposite, really, really small. It's microbes under the microscope. So each one of these tiny blue dots here is a microbe. However, I am studying the microbes to study outer space. So if that was your guess, you weren't entirely wrong. I'm growing them to study what sort of life could exist up there. I am particularly interested in one of Saturn's moons, shown here, called Enceladus. It has a cold and icy ocean and space geysers that gave us a window into detecting what its alien ocean contains. It turns out that it has all the right stuff to support this sort of life form. Hydrogen, its air, and carbon dioxide, its food. I hope that by studying these amazing microbes here on Earth, we can begin to learn what sort of life could exist beyond. Thank you.
All right, let's hear it for Sabrina. Thank you, Sabrina. That was great. Let's start off with you this time, Jorge. What did you think?
I thought it was spacey and great. And, pretty cool. I thought you were really confident and relaxed and really sort of sure of yourself in your delivery. And it was really interactive. I thought that was really fresh and fun, where you sort of, that time you pause to like have the audience think about something, or you asked a lot of questions of the audience. That was really good. And I also like that you talked about kind of what you did, like you specifically, and like I did this, or I'm studying this. I thought that was really cool to make it personal. And then the topic space, I mean, I wanted to hear more. I mean, alien life and alien environments. I thought that was really cool, and I wanted to hear more of that.
Great. Thanks, Jorge. Raven, what did you think?
I second what Jorge said, all of that, and just adding to the point that I feel like your confidence in your subject really made us want to hear more from you. So great job knowing your material. It was well presented and I enjoyed it.
Thank you.
Great. Thanks. Melissa, anything to add?
Yeah, they've both said it. It was just a great communication style. You know, you were relaxed, you were confident, you took great pauses. And I think it was really nice that you were able to connect tiny microbes here on our planet and connect them to outer space and the unknown, and get us really excited to figure out, okay, what's next? What are you going to discover next? So fantastic job.
Thanks.
Great. Thank you, judges. And thank you again, Sabrina. That was great. And a great job to all of our slammers. Now, to our judges, do you have any comments before you go off to deliberate? Jorge, how are you feeling?
I've been super impressed. I mean, you know, I've been to a lot of talks in my life. And just seeing the level of enthusiasm and engagement and really thought to like how you deliver the material was really high, the level, and really fun to be a part of.
Yeah, Melissa, how about you? Anything to share before you go off to deliberate?
It's going to be tough. I'm picking a winner. You all did absolutely amazing. This is why people do science communication, because it's passionate. It's us learning something new. I mean, you know, all of us here are experts in kind of our own right, and we're still learning new stuff from you guys. So you all did an amazing job, and you should be really proud of what you've done.
Great, thank you. Raven, anything to share before you go off?
This is going to be very hard. I know it's going to be a really tough decision for all of us to make. And please just know that everyone who presented today did a great job of, and just because you didn't win doesn't mean that your science communication is not good. We just had to pick one person, but I think you all are amazing. So keep up the great work.
Great, well, thank you so much.
Let's pick 6. Can we pick 6?
I know, right? Everyone's a winner tonight. Well, thank you so much, judges. It is now time. So off you go, and we will see you soon. Enjoy your deliberations. It's definitely going to be a very, very difficult decision. And those of you at home might be wondering how the judges are going to evaluate the slammers tonight. So we asked them to consider 4 judging criteria as they select their favorite slam to move on to the finals. And those criteria are scientific content, creativity, clarity, and one more very important element, their connection to the audience, you. Okay, now slammers, you all did such an incredible job. We're so proud of you. Our judges absolutely have their work cut out for them. And I would love to hear your thoughts before the audience voting begins. So would anyone like to share how you're feeling now that you have finally shared these slams that you've been working so hard on with our audience? Anything, any thoughts?
I wanted to say that it was an honor, like hearing every slam here. It's been really enlightening and really amazing. So congratulations to everyone. You've done amazing jobs.
Great, thanks for that. Any other thoughts about the competition before we launch the audience voting?
I was just going to say it was really great to see how everyone was able to share their research in the different ways that they did it. It was really great. Good job, everyone.
Definitely. All right.
I still feel very powered up from everybody's presentation that, yeah, excitement. Let's give another round of slams, everyone. It's going to be hard winding down from this.
I really enjoyed it also. I love learning about each and one of your presentations. And I think I speak for all of us. We just do this because we want the community to hear our science, to hear what we think, and to learn a little bit. While we have fun, will they have fun? Because I think that's the important thing of all of this, that we're able to give something to the community for them to use it in a positive way.
Wonderful. Well, that is a lovely thought to close on before we launch into the audience voting, which it is now time to begin. So here we go. Now it's your turn. To vote for your favorite Science Slammer, go to pollev.com/Museum of Science. That's POLLEV.com/Museum of Science and select their name from the options there. Or if you'd rather vote by text message, text Museum of Science to 22333 and then send us the number on the screen next to your favorite slammer. Those numbers are 1 for Anna, 2 for Giska, 3 for Miles, 4 for Kate, 5 for Carrie, and 6 for Jolene. The clock is counting down from 5 minutes, so make sure to get those votes in. And while the voting is going on, we'll be showing some of the comments that are coming in from our audience to cheer on our slammers. 2 minutes to go on the clock. Keep those votes coming. You can vote online by going to the web address on the screen, pollev.com/Museum of Science, or you can text Museum of Science to 22333 and send us the number next to your favorite slammer. Those numbers one more time are 1 for Anna, 2 for Giska, 3 for Miles, 4 for Kate, 5 for Carrie, and 6 for Jolene. Just 1 more minute to vote. And we are done. Let's tally up those votes. All right, audience voting has now ended, and we are ready to announce our audience choice winner. And the winner is Megan. You will be advancing to the finals on May 4th. Congratulations. I'm going to give her a second to come up here and join me. Hi, Megan. How are you feeling? Fantastic job.
Feeling very excited.
Great, wonderful. Well, congratulations. So let me ask you, what inspired you to enter the ReachOut Slams?
Yeah, I think that a lot of the time people think science is sort of hard to access because there are a lot of big words that a lot of people don't hear often. And I think the fun thing about slams is that they're targeted towards everyone, and that's how science should be.
Yeah, I agree. And so how did you come up with the idea for your slam? Where did that come from?
That's a good question. A lot of it, I talked a little bit about my project that I'm working on for my thesis. And so I've sort of spent a lot of time thinking about how these cells divide. And at some point, the maze idea kind of came to me. But yeah.
Great. Well, it was really creative. Thank you for that. And so you have, what you're studying is so interesting. This whole process of like cell division and how important it is that it's so accurate. What really excites you about doing this kind of work?
Yeah, I really love, so we use microscopes a lot, like you saw, and I showed that movie today of the cell dividing, and I really love doing science where I can like see stuff in action. It's really exciting to me to get to kind of like see things moving and watch the science happen in front of my eyes.
Oh, that is so cool. All right. Well, we are just going to give another minute. Our judges should be coming back to join us any minute now with their decision. It looks like they're having a tough time in there making a choice. So, Megan, I hope you're okay hanging out with me for a minute.
Yeah. I thought all the slams were so great, so it really makes sense that they're having a hard time.
Yeah, they were. What was something kind of interesting did you learn anything today from any of your slams?
I learned a ton of things. I had not thought too much about how learning about life here can tell us about life on space. So that was kind of like a, changed the way I think sort of moment. And I really love all the like relevant stuff we've heard about vaccines and antibiotics and stuff that feels very, relevant to our life right now.
Yeah, I think everybody did such a great job with really making everything relevant to us and really kind of bringing us into all these like interesting topics that I never even kind of knew existed, which is so cool. And it looks like our judges are back. That was a tough decision, I imagine. We'll give Raven just a second to join us again. And we will hear for our judges. There she is. Well, welcome back, judges. It sounds like that was a really, really tough decision, and I don't blame you because we had some great slammers tonight. So, Melissa, as our spokesperson for this evening, do you have any comments before you make your big announcement?
Yes. So first of all, you all did an amazing job. We had such a hard time picking a winner, and if we could have given it to all of you, we would have. You were just great with your communication styles and how you brought that passion and your knowledge through. So I just want to let you guys know we are all super proud of you. You should be proud of yourself because that was amazing. And yeah, great job. Thanks for making our job tough.
They certainly did. All right. Well, Melissa, who have you all chosen?
If I could have a drum roll, please. Is it happening?
Yes, it's happening. Who have you chosen to move on to the finals?
We have chosen Sabrina Elcasas to go forth. Congratulations, Sabrina. Really, really great talk. We absolutely loved your enthusiasm and how you got your message across. So great job.
Great job. Congratulations, Sabrina. How are you feeling? What are you thinking?
I'm feeling awesome. Thank you guys so much. Everyone's talks were so amazing. I feel very honored right now, and I feel like I wish everyone could have gone through because everyone did an amazing job.
I agree. That was great. Well, thank you. Congratulations, Sabrina. And congratulations to Sabrina and Megan. So you will each advance to the Reach Out Finals on May 4th with the chance to win our $1,000 grand prizes. And you will also be awarded $250 stipends to continue improving your entries for the finals. But really, congratulations to all of our slammers who presented tonight. You all did just an absolutely amazing job. Thank you so much. You will all receive $150 stipends to thank you for working so hard to share your science with all of us. So please, everyone, join me in thanking and congratulating all of our amazing slammers tonight. Thank you so much for giving us all some insight into your fascinating fields of science. And you know, one thing that really wowed me tonight was kind of learning about how all these different types of science can work together, like engineering and cell biology and microbiology and outer space and x-rays and vaccines. So cool. I learned so much tonight. And thank you to our incredible panel of judges who are all just complete rock stars and have generously shared their vast experience communicating science across the country, the world, online, on TV. Thank you so much. I'm going to give them all a chance to say goodbye.
Bye, everyone. You did a great job today. Congratulations.
It was really hard to pick a winner. You know, we all responded to different things, and we all loved each and every one of you. But we were all slammed for sure by science.
Definitely were. You all did an amazing job. You should be very proud of yourselves.
Yeah, definitely great competitors. It was really stiff competition here.
Yeah.
Wonderful. Thank you again so much to our judges. Thank you for being here tonight. And thanks so much to all of you for joining us tonight. And thank you to our sponsors, the National Science Foundation and the Center for Integrated Quantum Materials. Now, we would love to hear your thoughts about this event. If you would like to tell us what you think, please feel free to fill out a brief survey using the QR code that's up on the screen, or you can find the link at mos.org/reachout. We will randomly select 5 online audience members who complete the survey to receive a $50 Amazon gift card. And we hope to see you next week at the last round of the semifinals on April 20th, and then again for the big finals event on May 4th. You can visit mos.org/reachout to register. And congratulations to our winners tonight and to all of our amazing slammers. Thank you to all of them, our judges. Thank you to you, and have a wonderful evening. Good night.